So it's really my pleasure to introduce to you today Dr. Hume Stroud. He joined the Department of Neuroscience as an assistant professor in March of 2021, and he also has a joint appointment in the Green Center for Reproductive Biological Sciences. Hume is an endowed scholar. He's the W.W. Caruth Jr. Scholar in Biomedical Research at UT Southwestern. So Hume did his undergraduate degree at Cornell University, and then he switched coasts and went to UCLA to do his PhD, where he worked with Stephen Jacobson. Now, as a PhD student, Hume did something a little bit different. He studied epigenetics in plants in Arabidopsis, and he was extremely prolific as a graduate student. He was an author on 24 papers from his PhD studies, and these were all in terrific journals like Cell and Nature. And among those, he was first author on half of them. So 12 first author papers as a graduate student is very impressive. So then Hume went back to the East Coast. He joined Mike Greenberg's lab at Harvard Medical School in the Department of Neurobiology as a postdoc, and he shifted gears just a little bit. And he was still working in epigenetics, but this time in the mammalian nervous system. And I guess mammals are a little bit more challenging than plants. He only published 7 papers as a postdoc in Cell and Neuron. Actually, it was really impressive. He did an amazing job as a postdoc, studying what, building the foundation of what he's going to talk to you today. So we were really excited to recruit Hume. He's received several early career awards, including Simons Foundation Bridge to Independence Award, a Whitehall Foundation Award, a Brain Behavior Research Foundation Award, and a Klingenstein Award, among others. And as I already mentioned, he continues to study epigenetics, but at this time at the interface of neurodevelopment, and that's what he's going to talk about today.
Thank you, Jenna, for the introduction. It's really nice to have this opportunity to give everyone an overview about some of our research. About 1 in 5 people in the US suffer from some form of mental illness during their lives. Unfortunately, there are very limited treatment options for these disorders. Historically, proteins that function at the connection of neurons or synapses have been the focus in studying brain disorders. However, in the past decade or so, growing evidence suggests that regulators of chromatin, that is a DNA complex of DNA and proteins that make up chromosomes, are dysregulated in a variety of brain disorders. For example, for autism spectrum disorders, large-scale human genetic studies are finding that mutations in genes encoding for chromatin regulators are significantly overrepresented in people with autism. So these findings collectively suggest that dysregulation of chromatin may in part underlie a wide variety of neurological disorders. To understand how chromatin dysregulation leads to disease states, we really need to understand how chromatin normally works in the brain. Cytosine methylation is a key chromatin mark that occurs across the genome that plays essential roles in diverse biological processes, including X chromosome inactivation, genomic imprinting, and transposon silencing. In most mammalian genomes, DNA methylation occurs as CG sequences. So these are cytosines followed by guanines. And CG methylation at the promoters of genes is classically associated with gene silencing. Importantly, dysregulation of this mark can lead to embryonic lethality and disease states such as cancer, suggesting that this mark is essential for normal development and cell function. A really remarkable feature of the neuronal genome is the high presence of methylation that occurs at CA sites. So these are cytosines followed by adenines. About 10 years ago now, the labs of Joe Ecker and Bing Ren found that neurons have extremely high levels of CA methylation compared to any other cell type that's been looked at. In fact, in human brains, the amount of CA methylation can actually exceed the amount of canonical CG methylation, suggesting that in our brains, the dominant form of methylation is actually CA methylation. An interesting feature of this mark is that it accumulates across the genome, specifically in early postnatal life. This is the first few years of life, perhaps spanning up to 2 decades in humans, and in the first few weeks of life in the mouse. Intriguingly, this timing of CA methylation accumulation coincides with the timing of neuronal maturation and synapse formation, suggesting a potential role for this mark in these processes, although it has not yet been tested. I thought this was a really exciting discovery, and essentially nothing was understood about the regulation and function of this mark. So, for example, is CA methylation similar to CG methylation, or is it something different? Does it play roles in gene transcription? If so, is it a repressor or an activator? Why have neurons evolved to acquire this weird chemical mark across their genomes? So for my postdoc work, I decided to study this fascinating biological process. We found that the DNA methyltransferase DNMT3A is responsible for all CA methylation that happens in the genome, such that deletion of DNMT3A results in complete loss of this mark. DNMT3A is important for the brain in as much as deletion of DNMT3A in the mouse brain leads to severe behavioral defects, eventually leading to death. And in humans, heterozygous mutations in DNMT3A are found in individuals with autism and intellectual disabilities. So DNMT3 has very crucial roles for the brain. We found that DNMT3A transiently interacts with the neuronal genome, specifically in early postnatal life, and this time methylates CA sites in the genome. Once deposited, CA methylation persists throughout adulthood and in part functions to recruit the methyl DNA binding protein MECP2. MECP2 is mutated in individuals with Rett syndrome, a really devastating neurological disorder that's probably the most severe form of autism spectrum disorder. Once bound to CA methylation, MECP2 appears to function to restrain transcription of many key neuronal genes across the genome. So I think, or at least I like to think, that we made progress in understanding this CA methylation pathway. There are actually many open questions. For example, how do neurons acquire CA methylation? I think this is an interesting question because DNMT3A is actually expressed in all cell types in the body and not just in neurons. And in these cell types, DNMT3A methylates CG sites and not CA sites. Moreover, the intrinsic preference for DNMT3A is to methylate CG sites. So here we incubated DNMT3A protein with unmethylated DNA in a test tube and quantified its methyltransferase activity. And what you can see is that DNMT3A by far prefers to methylate CG sites over CA sites. So given this intrinsic property of DNMT3A to methylate CG sites, how do neurons acquire such high levels of CA methylation? That is, how does the same enzyme DNMT3A methylate CA sites in neurons, but methylate CG sites in most other cells? There must be some unique regulation of neuronal DNMT3A that enables the enzyme to methylate CA sites. So we're very interested in understanding how DNMT3A behaves differently in neurons. And we're exploring the mechanisms by which this switch in enzymatic activity occurs in vivo. We're also very interested in understanding the methylation-independent roles of DNMT. So for about 25 years since its discovery, DNMT3A has thought to be primarily an enzyme that methylates the genome. However, we recently obtained unexpected evidence that DNMT3A likely plays roles outside of DNA methylation as well. So we're putting in a lot of effort into trying to understand these methylation-independent functions of DNMT proteins. We're also interested in understanding the function of CA methylation. To understand the function of CA methylation, we need to identify the proteins that bind to this mark. As I mentioned, we and others, including the labs of Huda Zogby and Adrian Bird and Hongjun Song, have found that the methyl DNA binding protein MECP2 tightly binds to CA methylation. However, genetic evidence suggests that there are likely other readers of this mark. Excitingly, through proteomic approaches, we recently discovered a number of putative CA methylation binding proteins that we're actively investigating. And my hope is that by characterizing these CA methylation readers, will provide insights into the roles of CA methylation in the nervous system. Finally, we're exploring the link between the environment and the neural epigenome, including DNA methylation. Although it's very clear that changes in the environment, for example, through exercise or diet, can affect our brains and our behaviors, there's actually very limited mechanistic understanding on how these specific environmental cues can modulate the brain. Interestingly, most chromatin enzymes, including DNMT3A that puts on methylation, and TET enzymes that facilitate the removal of methylation, depend on specific metabolites to function. So we're testing whether physiological changes in specific metabolites can alter animal behavior by modulating the neural epigenome. So our hope is that by studying these interesting chemical tags that decorate the genome will provide insights into how the brain works and will ultimately provide insights into neurological disorders and potential therapeutic strategies. These projects are led by these fantastic scientists I have had the pleasure to work with. And I'm also very grateful for all the support from colleagues throughout UT Southwestern, especially in the departments of neuroscience and the Green Center. And thank you for your attention.
Question? Okay, we have a question there.
Thank you. If you go back in evolution, when do you see CA methylation prevention in neurons?
Yeah.
And can that be a guide for your molecular search?
Yeah, so profiling work by Ryan Lister's lab have found recently that the CA methylation mark is found in neurons in vertebrates. It's not found in insects and invertebrates. So it's something that happened in evolution in vertebrate lineage, but I agree it's a way to try to figure out how this process works, but we still don't know.
Okay, so your current model on how this metal transfers have different specificity is what different factors or different cofactors? Do you have any candidate?
Yeah, so we don't have a definitive answer, but our work in progress suggests that a specific phosphorylation site on the DNMT3A enzyme that might specifically occur in neurons may facilitate the switch in enzymatic activity from CG sites to CA sites. And we're still trying to figure out this pathway.
Thank you. Okay, we have one more question before the break.
So Hume, it's interesting that the CA appears over a time frame that correlates with maturation of the brain. Is that activity dependent CA appearance, or is it developmentally regulated independent of activity?
Yeah, so still, honestly, that's a little bit unclear. We have some evidence suggesting that it might be partially activity dependent. So this phosphorylation site on DNMT3A, we think is activity dependent. But we're struggling to really distinguish if it's intrinsic or activity dependent. But I think that's an important question that we're trying to figure out.
Thank you.
Yeah, thank you.